Good afternoon, everyone. Thank you for attending our webinar this afternoon. My name is Lisa Roberts, and I'm with the Florida Wildflower Foundation. First, I wanted to let you know that we'll be recording this session, so if you only can attend part of it or would like to hear it again, we will be sending you the link hopefully in about 24 hours after we end the session. You can always view our presentations on our YouTube channel, and that's youtube.com/FLA Wildflowers. We also invite you to email us at info@flawildflowers.org should your question at the end of the presentation not be answered. We know we're probably going to have a lot of questions. We try to get to as many as we can, but sometimes we don't get to them all. So feel free to email us. If you are new to our organization, we protect, connect, and expand native wildflower habitat throughout the state. And we do that through our education, research, planning, and conservation programs. You can learn more about Florida Wildflowers and our organization's work at flawildflowers.org. Our funding primarily comes from the state wildflower license plate, and whether you have the old look or the new one that you're seeing here, you are helping to fund research, education, and planting projects statewide. And that is because each time you purchase or renew this license plate, $15 comes to our organization to fund our work. Now I'd like to introduce you to our presenters. This is Small Space, Big Benefit, and they are all from Stetson University in DeLand. First, we have Karen Cole. She directs the Gillespie Museum, which is Stetson's Earth Science Museum. She has taught and published on environmental writing and education in natural areas. And Cindy Bennington is a plant ecologist whose research interests include plant adaptation to stressful environments, natural science education in situ, and plant and pollinator interactions. And then we have Peter May, who publishes on ecology and the behavior of reptiles. A published photographer, he also regularly posts his significant sightings, field reports, and other bits of Florida natural history on Facebook. I'd like to thank you all for joining us, and right now I will turn everything over to Karen.
So Lisa, thank you so much for this opportunity to talk today about the Volusia Sandhill Ecosystem. As you mentioned, it's a teaching landscape and now a research site at the Gillespie Museum on the Stetson University campus. We're actually approaching our 10th year of this urban restoration project, which is basically a 1-acre area on the southeast corner of the campus, the DeLand campus. So this afternoon, I'm going to talk for a few minutes about the timeline we have had for developing this longleaf pine, because we think there's some interesting lessons for wildflower establishment built into that story. And then I'll turn things back over to Cindy, who's going to talk about hers and Peter's research on pollinators. And it, of course, the research that they've done really points to some significant benefits of small fragments of native wildflowers. And it's a message we know that we share with the Florida Wildflower Foundation. And we really appreciate the way in which the foundation's been amplifying this message through its programming and grant support. The Sandhill Ecosystem really has gone beyond its original conception as a demonstration of an historic landscape, and now it provides field experience and research opportunities for undergraduates and faculty, as well as some measurable ecosystem benefits. The museum is the university's Earth Science Museum, as Lisa mentioned. It was founded in 1958 with a gift from a couple of Central Floridians, Thomas Bird and Nellie Gillespie, who donated their collection of about 15,000 mineral specimens and a nice endowment to house and support the museum. It's a very small academic museum. It's housed in a former residence on the edge of campus. And it has a mission to provide environmental education in Central Florida. So the Volusia Sandhill Ecosystem is one of a number of its initiatives. So in a typical year, which is certainly not this one, we're still closed to visitors at the moment, the museum welcomes thousands of visitors and residents and tourists, and a lot of K through 12 classes for field trip experiences, where our visitors can see displays of museums and special exhibits, and we support a lot of evening and weekend environmental programming as well, all of them centered on this historic collection of minerals. But we think of the museum really as one with and without walls. And in this photograph, you can see a visitor on a Saturday morning at one of our Science Saturdays, who's in our Florida Formations Gallery. But the important thing besides those beautiful specimens is to look out the window, which open to the native landscape on the museum grounds. 20 years ago, the museum staff and importantly, faculty from environmental and biological science developed the Florida native landscape as the museum's first outdoor classroom. A demonstration landscape, it inspired a native plant initiative on the campus and has served as one of the founders has nicely phrased it, it served as a reminder of what was and what could be, again, a place where patience and close inspection continually reward us with the subtle beauty of natural Florida. In the photograph, the bottom, you see two of our interns who are maintaining plant identification signs for our landscape tour of the 320 or so well-established trees and shrubs, which represent 52 species native to Florida. So in this small arboretum, which surrounds the residence, the older residence building of the museum, you can see plants that for at least 20, now going almost on 25 years, have flourished without fertilizers, biocides, or any significant irrigation. So the purpose of this early project was to introduce the beauty of the Florida landscape and to encourage the use of native plants in home and commercial landscapes. 10 years ago, this Brinker Environmental Learning Center opened on the grounds. The classroom building and the surrounding area offer a chance for us to move to an even more site-specific native plant restoration, which is the Volusia Sandhill teaching landscape. And that landscape is on a 1-acre plot just behind this building. So this is sort of helping get you oriented. Much of present-day DeLand was once longleaf pine, Sandhill Habitat, which is one of the most extensive woodland ecosystems in North America. I'll remind you of a story that you as wildflower lovers know too well, that beginning in the 19th century, longleaf pine forests were felled for timber and turpentine operations. Later, Sandhill Habitat was cleared to make way for citrus groves, and of course now for golf courses and housing developments. These two photographs tell the story. On the left is on my left is an 1880s archival photograph of the first campus building, DeLand Hall, which shows that in its early days, the Stetson campus was still surrounded by a longleaf canopy. And then the more recent photograph from 2016 shows the palms and live oaks that now cover the campus and much of the city of DeLand. So as a connection to our Florida formations room that exhibit in the museum, and a way to connect our geologic roots with the outdoor museum, you can see here a little bit about, just to remind you about the Sandhill Habitat. The dry sandy soils of this habitat were of course formed as dunes when sea levels were higher. Sandhill sites are found in uplands throughout the southeastern United States. And the dominant tree, which you can see in this wonderful photograph of a mature site, they're dominated by longleaf pine, Pinus palustris. Cindy and Peter are going to talk a little bit more about this ecosystem as they discuss their pollinator research. But as you can see from that, the photo, besides this being an historic landscape for our area, that open canopy, even in the most mature landscapes, allows for a highly diverse understory of grasses and perennials. And so it's a very nice place for establishing wildflowers. So here's where we started. These photos are from an Arbor Day celebration in April of 2011. A small group of students are gathered behind the Rinker Environmental Learning Center on our sandy hill there, you see, and they're planting the first two longleaf pines that started in the whole project. So this is the moment of genesis for the project. With grant support from Volusia County Department of Environmental Management, we were able to begin really the first phase of the reforestation, helping us establish, the funding helped us establish a temporary irrigation system and to purchase 80 trees, longleaf, some turkey oaks, persimmons, and black cherries that provide the overstory and really visually defined the teaching landscape initially. So here we are on the south entrance of the landscape. So for the first few years, the passersby really saw those 80 or so trees. And if they went to our website, they would have seen a kind of aspirational landscape plan. This photo and a number of others that you'll see are really taken from the bottom of our sandhill, from what's called the DeLand Greenway, which is a joint project between the city and county, establishing a pedestrian and bicycle path. So this is fairly early in the project. By September of 2011, as you see, the trees were doing well, and also a lot of uninvited turf grass. And in the following 2 academic years, with volunteer support from the community and from undergraduate classes, we were able to keep the irrigation repaired mostly, and planted and protected some plots of wiregrass, Aristida stricta, which is of course the dominant ground cover for mature longleaf areas. Those are not so easy to see from this photograph, but we did begin with, as an understory, planting a number of plots of wiregrass. And in those years between 2011 and 2013, we also developed the understory closest to the Rinker Environmental Learning Center, where the undergraduates gathered for classes and all of our museum programming was staged. So really divide and conquer was our strategy. We would use small groups of volunteers or students in usually 2 to 3 hour work days, work afternoons, to begin to form really small plots and replant. By the summer of 2014, the trees were well established, and we were really ready to expand our understory planting. And this was a big turning point for us. So with grant funding from the Florida Wildflower Foundation, that understory began to take shape. In the fall months of 2014, the southern entrance, which we've been looking often, of the teaching landscape became one of a number of Viva Florida demonstration gardens throughout the state. The grant allowed us to prepare and plant a demonstration site of roughly 500 square meters. So a much larger swath of the landscape we were able to plant in that area, now bordering the DeLand Greenway. So the southern entrance became much more beautiful from that Greenway path. Here you see some of the student volunteers who helped us get going. The one at the bottom is standing in one of the seed beds that we mapped out with orange and identified as we were waiting for the seeds to come along. So in that series of work days, usually on Friday afternoons, for about 8 weeks in the fall of 2014, the site was cleared and planted, seeded, mulched, 200 or so 250 plants and thousands of seeds from the Florida Wildflower Growers Cooperative were included. And you'll see in that upper left-hand corner some folks you might know. So this was a big moment for us, and we've always been very grateful to the Florida Wildflower Foundation for that early support. In the late summer and early fall of the same year, and partly because we had made such a visible change in the landscape, we were able to get some more support from our facilities friends who helped to install walkways through the landscape, and we also included interpretive signs. So it made space for more types of informal education. The 8 interpretive signs at the moment that we have along the walking path create teaching stations and provide information about longleaf pine forest and the wildlife dependent on them. Here's a photo, one of Peter's, from 6 years after the first plants were put into place. So you can see how things have developed. It's not been all glory. This is a photograph from roughly the same place, but in the fall of 2018, we had a major setback with, after an active hurricane season, we had 2 pine sawfly infestations, which killed more than a dozen trees, and unfortunately some of the largest trees that we had in the site. But we're recovering. We've planted new trees and some additional turkey oaks. We've expanded grassy areas. We now have a legume garden, you might say. And as you can see from this photograph, the understory is coming along especially well. So at this point, I'm going to turn things over to Cindy to talk about how those understory plants have contributed, how they've used them in their research.
Great, thanks, Karen. Can everybody hear me? Am I?
Yes.
Okay, great. So yeah, that developing understory brought with it insects. I mean, we could literally hear the buzz as we started walking around, as we were getting more flowering plants. And that really stimulated our interest in developing a baseline. Well, what's there? What are the insects that are present? And that initial interest in simply documenting the insect visitors, combined with our understanding that pollinator populations have been declining globally, is really what led Peter and I to into what has become a multi-year research project. And if you were here from the beginning, you know that Peter is present on this Zoom, and he'll answer some questions after the talk, but I will act as the presenter. And he is definitely the insect person. So if you have questions about that, he's the man to ask. Also, if you have questions about his incredible photography, these are all photos that he has taken. And I just have to get back, there we go. So declines in native pollinator abundance and diversity have been attributed to a number of different causes, with loss of habitat being a noted cause in many cases. A wealth of recent literature, interestingly, largely coming out of Europe, has suggested that small habitat fragments in urban settings can help maintain pollinator populations. And in some cases, those fragments can be as small as your backyard garden. So our goal in our project was mainly focused on our campus fragment, our Volusia Sandhill ecosystem. And we wanted to know whether the insect pollinator diversity and abundance that we were seeing there, how did that compare to a larger, more natural sandhill fragment? And we found that comparison site, that comparison fragment, in an upland area of Hart Island Conservation Area.
Cindy, somehow you muted. I'm sorry.
I don't know. Can you hear me now?
Yes.
We can hear you. Yeah.
Sorry, I don't know what happened. That's weird. I didn't touch mine. So I don't know how long I was talking. That's probably okay.
You were about to tell us about Hart Island.
Okay. Oh, great. Okay. So I didn't babble for too long with no audience. Yes. So Hart Island Conservation Area is this place that's managed by St. John's River Water Management District. It's actually a very large area, and there's a small portion of it, relatively small portion, that is upland sandhill habitat. And the St. John's River Water Management District very generously gave us access to this site. So this was our beautiful drive into the site on summer mornings. So these are aerial photos of the two sites, our campus site in the bottom right, and then the Hart Island Conservation Area in the top left. And you can see from those aerial photos that there are differences in the extent and urbanization of neighboring habitat. Those things are obvious, but they also happen, they're similar in that they occur on the same sandy ridge, those ridges that Karen was talking about. This is the DeLand Crescent City Ridge, where the DeLand campus is where that red star is. Hart Island is where that blue star is. And these are about 12 miles apart, so not too far apart. And we wanted, when we started our study, we wanted to control for differences in the total area we surveyed. So we restricted our surveys at Hart Island to that area that's within the black rectangle there in that upper left aerial photograph. And I put these abbreviations there because you'll see, I'll show you some data in a minute, and we abbreviate Volusia Sandhill as VS and Hart Island as HI. So just remember that VS is the campus site, the small urban fragment, and Hart Island, or HI, is the larger, more natural area. So once we found a comparison site and we felt like we had something to compare the progress of our sandhill to, we needed to settle on some research methods. How are we going to go about quantifying the insect pollinator abundance and diversity in those two sites? So in the end, we decided to restrict our observations to the insects that were visiting two weedy annual plant species that were present at both sites. Partly because they were present at both sites, relatively abundantly, and also because they represented two rather different pollination strategies. Biden's alba, the one you see there on the left, attracts a lot of generalist pollinators, everything from beetles and flies and bees and butterflies to, all of those things. Whereas partridge pea is exclusively pollinated by bees and wasps, but mainly by bumblebees. It's buzz pollinated, and so to release the pollen from the anthers, the bee has to get in there and vibrate its flight muscles at exactly the right frequency to get that pollen to be released. We use the same methodology for both of our plant species by going out every week in the summer from May through August, and we would visit both sites. We always went to Volusia Sandhill on Tuesdays and Hart Island on Thursdays. And while we were there, we made 10 5-minute observations to each of the species. In 3 years of data from 2016 to 2018, we recorded more than 5,000 insect visits from about 100 hours of observation and more than 30 different species of insects. We've continued those observations in 2019 and 2020, but today we're reporting only on the data we analyzed for a manuscript that was recently published in Natural Areas Journal. And I think there's a link to that URL. You could see the abstract in the chat, or if not, we have the link at the end as well. So one of the things we wanted to do was determine whether the composition of the pollinator community was similar between sites. And so for that, we used just the data from Biden's alba, because remember, that's the plant species that attracts a diversity of generalist pollinators. So on this graph, each of the 6 bars you see represents the total visitation to Biden's flowers at a particular site in a given year. And the different fill patterns represent different groups of insects. So for example, the dark solid black is wild hymenopterans, so wild bees and wasps. And I say wild there, meaning everything we observed except European honeybees. So we made those a separate category. You see that here, and it has kind of a cross hatching. So that's this color bar. Lepidoptera, butterflies and moths, they have the kind of scale pattern. And then flies are this sort of, I want to say, a screen pattern. And then coleoptera is the dots. So there's a lot going on in that graph. I just want to focus your attention on 2 main conclusions. First, if you just look at this solid black bar, you see quite obviously that native hymenopterans were proportionally more frequent visitors to Biden's alba in the Volusia Sandhill compared to Hart Island. So this small campus fragment had more visitation by hymenopterans proportionally than did Hart Island. And then secondly, lepidopterans, that's this group, were proportionally far more frequent visitors to Biden's flowers at Hart Island compared to Volusia Sandhill. So that suggested that the composition of the communities differed in some important ways. Bees were the predominant pollinator in our small fragment, whereas bees and butterflies were almost equally frequent at Hart Island. So in addition to kind of looking at the community composition, we wanted to look at, ask a question about pollinator abundance. We expected that the more immature understory of the Volusia Sandhill might not attract as many pollinators as the more mature understory at Hart Island. So we used our observation data to calculate visitation rates by insect pollinators to both species. The density of open flowers, both on our target species, partridge pea and Spanish needles or Biden's alba, differed between sites, as did the non-target species. There were just generally on any given day far more open flowers at the Volusia Sandhill than Hart Island. And you can see this is just an example just for partridge pea, but in the Volusia Sandhill, plants tended to be bigger and have many more flowers than at Hart Island. So because of that, and we wanted to make comparisons between sites, we calculated visitation rate as the number of visitors per flower per minute. So we were sort of standardizing across those differences. And in the next 3 graphs, I'll show you the y-axis, or the dependent variable, is visitation rate, that number of visits per flower per minute. So for this one, this is just for Biden's alba, just visitors to Biden's alba, but it combines all visitors. And the main takeaway here, you could see the lighter gray bars represent what's going on at Volusia Sandhill. The darker gray is Hart Island. And this is in all 3 years. There tended to be slightly more visits to the flowers of Biden's at Volusia Sandhill compared to Hart Island. In this next graph, we break that data out into the 4 main orders of insects that we observed as visitors. So in the top left, these are our wild hymenopterans, wild bees and wasps, so we excluded European honeybees. Lepidopterans, butterflies and moths, Coleoptera, the beetles, and then dipterans, the flies. So again, I'll just draw your attention to some major differences here. Native bees and wasps had far more visits to Biden's alba in the Volusia Sandhill compared to Hart Island, whereas butterflies and moths made more visits at Hart Island, and beetles made more visits at Hart Island. So again, this is sort of illustrates that community difference between sites. There's a lot of visitation going on at the Volusia Sandhill, but it tends to be made much more by bees relative to Hart Island. And for flies, it was about the same between sites. And then this final graph is just the only one we have for partridge pea. Remember, this is the plant that's pollinated or visited mainly by bumblebees. About 85% of the visits we observed to partridge pea was made by a species or another of bombus or bumblebees. And what you see here is kind of consistent with what we saw on Biden's, and that is that visitation rate tended to be higher in Volusia Sandhill. When you do the statistics, it's not statistically greater, but in every year there was a slightly higher visitation rate to partridge pea in the Volusia Sandhill compared to Hart Island. So the conclusions we draw from this, based on our data and that of other similar studies, make it pretty clear that small urban habitat fragments can sustain abundant and diverse pollinator communities. But the exact pollinator community composition might not mimic that which you would find in larger, more natural fragments. So just to circle back around where we started with Karen, the title of our talk was Small Space, Big Benefits. And this was a teaching landscape that we started with the idea that we wanted to educate visitors about the historic landscape that once existed on this DeLand upland ridge. But it's become so much more than that. It's become a place for research for faculty and students. It's become a place for community involvement. And thanks to the support from groups like the Florida Wildflower Foundation, a place where we can showcase native wildflowers that are appropriate for home gardens. And toward that end, we've done things like we've had students in various classes propagate plants for giveaways at some of the Science Saturday events that are hosted by the Gillespie Museum. The Gillespie Museum also houses a seed library that's includes things like vegetables, but also we've started including native plants. So students harvest some of the seeds from the site and then make them available to visitors to get through the museum. We've hosted outdoor classrooms for families and K through 12 visitors to learn about the Sandhill, not just the ecosystem, but also, plant reproduction and plant pollination. And it's also given us a way to teach undergraduates how to teach and how to express concepts that might be difficult and scientific and interesting, but express them to a younger audience in a way that's clear. And even in this age of COVID, the site is an important teaching space on our campus where students can be outdoors stay socially distant and still learn something about the natural world. So we've learned some things by creating this space, in addition to learning some scientific facts about pollination, which have been fascinating for me and Peter, but we've also learned a lot about just how to create this kind of space. And Karen had talked about that whole history of the development of the site and mentioned that, you know, there are challenges using these kind of disrupted landscapes, landscapes that have huge human impacts prior to any sort of restoration. We found that the soil there was filled with weed species. Surprise, surprise, that doesn't really surprise anybody probably who's tried to do this in their own yard or in a small patch. And they're not always weeds that you want. They're often not, some weeds we do like, but they're not always weeds you want, and they're not always native. In urban settings, you're surrounded by cultivated areas where there's more sources of exotic seeds and other unwanted things from your garden. And then there are things like sawfly infestations, which we definitely did not see coming. And they're sort of inevitable, and you just have to go with it and you know, try to find a way around it and try to find a way forward. So having some realistic goals and a good attitude, again, what anybody would think about their own garden. As Karen mentioned, we really restricted our intense efforts to small areas at any given time. It's very satisfying that way. So especially when you're working with volunteers or students, giving them a chance to actually complete something rather than just work willy-nilly on small spots was very important. And we found for our own sort of mental health and our own ability to appreciate the site, it's very helpful to feel good about a certain area at a given time. We've embraced some of the weedy stuff, Biden's alba being a good example. Many of you are probably familiar with that plant. We have sort of a love-hate relationship with it, which I think is probably pretty common for anybody who's familiar with it. It can take over, but it is an awesome pollinator plant. And while it's not certainly specifically limited to sandhill sites, it can grow almost anywhere, and it certainly has its place. And, you know, maintain, maintain, maintain. We pull a lot of weeds, and that's okay. We're keeping it together. So Karen's going to come back to just take us out with a few final acknowledgements, and then we're available for questions.
So I think that that Stacy Matraz is helping us post some of these resources that are available, including the link to the longer full study that Cindy and Peter have just recently published. Cindy and I did a project a few years back on the first 5 years of the Sandhill, Volusia Sandhill Ecosystem, and for a special issue in the Southeastern Naturalist, focusing on using sites in situ, natural areas for teaching undergraduates and younger scientists visiting the area. If you're interested in the full plant list for the Volusia Sandhill, that is, there's a link to that on our Volusia Sandhill website, and the Volusia Sandhill Ecosystem web pages are linked there as well. We had quite a few Sandhill ecosystem and ecology resources in place before March, but as we have all been shuttered and working remotely, particularly our undergraduate museum guides and our Sandhill interns have helped to contribute quite a number of interesting resources on those topics, and those are listed on our web page as well. And I want to mention that we do have a Facebook page, and we post weekly, Wildflower Wednesday, postings of things going on, interesting sightings and blooms, and sometimes visitations, surprising visitations to the Volusia Sandhill. So that link is there as well. I hope Stacy will be able to get that there. And this is a short list of the many, many organizations we have from whom we've benefited. Of course, we mentioned the Florida Wildflower Foundation grant, which was such an important moment in the development. The County of Volusia, through grant work, through permissions, along with the St. Johns River Water Management District, have been very supportive. Our Stetson University Facilities Management Group has always stepped in to help when we have needed it. Most of the work done in the Sandhill, particularly the understory work, is volunteer-driven, student-driven, but facilities colleagues are always ready to help us with some of the larger projects, from pest management to putting trees in the ground when it's necessary. So we're grateful for that cooperation. And the museum and the Sandhill are really part of the university, but specifically the College of Arts and Sciences. And we've always had tremendous support from that college, and are grateful. So I think that is the end of our presentation, formal presentation, and we'll be ready for questions. And we'll hope to have some answers.
Thank you so much, Karen, Cindy, and Peter. This has been great, and it's been especially rewarding to see the Sandhill develop over the years. It's turned out to be such a wonderful project and site. Stacy, I'm going to turn it over to you.
Okay, can you guys hear me okay?
Mm-hmm.
Okay.
We can.
We've got quite a few questions. Try and organize these a little bit because there's some overlap. Well, first of all, people want to know which wildflowers are best to plant now for pollinators that will bloom and endure through the winter in Central Florida, zone 9.
Wow, that's a hard question. You guys probably know the answer to that better than I do. I mean, now's not a great time to plant stuff unless you're going to water it for sure, right? But there are certainly things that overwinter, but I don't know. I mean, it's if you have a seedling or an adult plant, I don't know. I mean, stuff would be fine. You could plant almost anything that you could find at a native nursery right now, as long as you kept watering it. Something like liatris, gay feather would still go in fine, I think, and would flower next year, because they wouldn't flower until late summer anyway, or goldenrod would be okay. Those would be good pollinator plants, I think.
Bidens will bloom all winter.
Vines will bloom, yeah. You don't really have to plant that, but yeah, vines would bloom.
Your Conradina's going nuts right now.
Yeah,
So
Conradina is very good. That's a really good one. Kind of shrubby. You'd have to have like if you wanted it to be flowering through the fall, you'd have to start with a large plant, or plan on not having it this year. But yeah, that's a really good one.
I would also say, and I'm sure that Lisa and Stacy will want to weigh in on this question too, but I would also say that you could plant now and maybe begin to see rewards about this time next year.
Right.
So it's a long game in a way. So the plants would do very well through the winter, particularly with some water, but actually beginning to see the blooms and the pollination interaction could take a bit longer.
Yeah.
And we have a few questions about site preparation. How much preparation did you do? How did you get rid of the turf grass? And can you recommend any herbicide that targets turf but is safe around honeybees and gopher tortoises? If you used herbicide.
I guess I can answer that. Karen, you want to just chime in? But so we did a variety of things for site prep. For the Florida Wildflower Demonstration Garden, which was the biggest area we ever did at one go, we had professional application of Roundup. That wasn't our original intention, but that's how it ended up going down. We haven't, I mean, if we use a herbicide, it's always Roundup now, and it's always very very carefully applied. In other words, just very spotty. We don't do any broad scale application of any sort of herbicide. The only time we did that was in preparation for that large spot. But we've used a number of different things. We've used a lot of solarization for some of our other smaller spots. So especially in the summer, if we put down clear plastic in June or July and just leave it for a couple months, that does a pretty good job of killing turf grass. You know, nothing keeps it away forever. After we've done that, after we've gotten rid of it through solarization, for example, we've used a combination of things like paper and landscape cloth to keep the weeds back, and then applied lots and lots of pine straw as a mulch to try to keep grass back. And like in that area where we started close to the building, that's the area where we've probably been the most diligent about continuing to weed and keeping mulched. And I'd say now we have a really good handle on the grass and kind of the unwanted weeds. And it's not like there's never anything, but that's pretty good. Other places, it's just very hard. Grass comes back. Mowers go right by and blow grass seed back in, that, you know, when we get rid of it. So it is really hard to do. But I think with some people use cardboard, like instead of just newspaper or paper, they'll put cardboard down and landscape cloth on top of that. You can do a pretty good job. We've tried stuff like vinegar, horticultural vinegar. It works okay, but not great. It's hard to get rid of grass.
Okay, did Hart Island have the same devastation as the Volusia Sandhill during the period with the disease that hurt your pine trees?
No, not at all. I never saw a sawfly at Hart Island. No.
I saw them one summer. I saw one or two plants attacked, but they never went beyond that. It never turned into an infestation like it did on campus.
Yeah.
Do you have any thoughts on why that would vary so much between the two sites?
We've talked to a bunch of folks about it, and a couple things we heard. So one was that year, 2018, was a bad year for sawflies, and that they tend to have sort of a 6 or 7 year cycle, and sort of county-wide it was a bad year. But also that they prefer younger trees. So not only did we have younger trees, but we had younger trees that weren't surrounded by any other trees really. So I think once the sawflies got there, you know, they kind of just devastated the few trees that were there, because it's not like there's a whole bunch of other longleaf they could have gone to. There's another place not too far from campus that had a bunch of young longleaf, and I saw those got nailed really badly too at the same time. So I think that the age of the tree might have been really important. So one of the things I said near the end was, you know, setbacks can be challenges. So one of the bright spots we took from this is we started with trees in 2011 that were all approximately near the same size, and we've added a few more since then. But this was an opportunity to really switch it up and get some real age structure to our forest. So now we have some really young ones coming along in addition to the ones that are 10 years old. So we've made lemonade out of our lemons. But so hopefully if these guys get, you know, another 10 years from now, 6, 7 years from now, whatever, as they get larger, if there were another infestation as bad as that one, that it wouldn't necessarily kill the trees. And we had some that were totally defoliated and did not die. And then we had some that were defoliated twice, because we had a big infestation in August of that year and then October. And we had some trees that were hit both times and didn't die, and others that were only hit in the last time and did die. And we really never could figure out what the difference was.
Would regular irrigation at Volusia Sandhill explain the greater abundance or the number of flowers compared to Hart Island? Does that have any effect on what you saw?
Yeah, we don't really irrigate there anymore, but the big difference is nutrient levels. So we've had the soil tested in both sites, and when we did irrigate in the Volusia Sandhill, we irrigated with gray water, plus the soil that's in there is mostly fill from God knows where. And nutrient levels like nitrogen, phosphorus, all magnesium, calcium are all way, way higher, like orders of magnitude higher in the Volusia Sandhill compared to Hart Island. So it's very unnatural. I mean, the low nutrient, low pH soils of a sandhill are absolutely not present in the Volusia Sandhill, where we have very high pH, high nutrient soils.
We also have a swimming pool buried under the sandhill. So.
Yes, yes. which puts a dent in drainage sometimes. Yes.
Yeah, it reminds you of some of the unusual aspects of our geology out here.
Yes.
Were there any species or groups of perennials that stood out for pollinators?
Peter, what would you say?
Nothing's popping to mind.
Yeah.
I mean,
Go ahead.
there are some species that sort of explode, like Pediopsis. We've got a lot of Pediopsis that towards the end of the summer and fall. And so the stuff that's most abundant is what gets most of the visits, like what was it? You got, drawing a total blank now.
Goldenrod? No.
The Penstemon that was blooming.
Oh, yes. Yeah. Penstemon.
You know, this year I had a couple of plants of Penstemon that put out huge showy displays, and they were being visited by bees pretty heavily. So it all depends on, you know, what's there.
Oh, yeah. And like Monarda, bee balm, that gets a lot of visitation. That's pretty easy one to grow too. A little bit weedy looking, but.
If you want to get a lot of visitors, plant some Phylla. Phylla notiflora.
Yes. Yeah. Yeah. Fog fruit. Yep.
There are some plants in the sandhill that are fire dependent. How did you handle that need? Or how are you handling that need?
Not with fire at the moment. So maybe the question refers to things like wiregrass, which typically doesn't flower without fire, for example. And we're certainly aware of that. We've talked to lots of folks about ways to perhaps introduce a very small fire, or even like to wiregrass, just individual plant fires. But we've also used a weed eater on wiregrass and gotten it to flower. So at the moment, Karen's probably better to address this than I am, but you know, as she likes to remind people when they ask us about fire, like what would come back right now if we burned is a lot of weeds.
Yeah.
Yeah.
We would be back at our starting point. So for wiregrass, besides just whacking it with the weed whacker, we've also tried just severe pruning. We've had a few students experiment with just burning leaves and then putting that around the base of some wiregrass. But the truth is the severe pruning for those, for that species especially, seems to work as well as anything. It's more fun to get out the torch, but
we do have, in just the last few minutes, we have quite a few questions on the solarization process, whether or not it's destroying the seed bank or the soil microorganisms, how you're managing the dead material once you solarize. Can you address that briefly?
Yeah, I would say solarization well, we don't have a seed bank except for a seed bank we don't want. So if solarization destroyed our seed bank, it really wouldn't, in the places we've used it, like now that we are starting to build up a seed bank in certain places, we wouldn't do solarization. We're doing a place where it's pretty much straight up lawn. And the only thing in the seed bank there is either more lawn or weeds that we don't want. So that's not really an issue. Now, microorganisms, potentially, I think one thing we do that in probably a good way is by concentrating on small areas at a time, we're not, microorganisms can move pretty quickly. So yeah, maybe we're locally affecting microorganisms for a brief period of time, but it's a couple months when the plastic is on and then we take it off. And I feel like it would not take long at all for that microbe community to build back up. What do we do with the dead stuff? We make a big pile, and then Karen calls facilities and says, come get our big pile of stuff.
Okay, and did you happen to notice any or take note of any ants in either of the sites? Do you document those as pollinators?
No.
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 I mean, there are ants there, but they're not frequent flower visitors.
Okay. Sorry, I'm trying to answer some questions while I'm reading the questions. I apologize. There are a lot of questions here. We do have a couple questions about the site as far as visiting it. It is open to the public right now, correct? The outdoor area?
Unofficially, it is. The campus is still not completely open to visitors, but we're on a corner and frequently people walk through our corner on our paths. We actually leave material both in a little box that's called the pollination station and on brochures on the back porch of the Rinker Environmental Learning Center, knowing that everyone needs a place to walk and to browse the sandhill. So it is open, and I don't think you would be challenged by the public safety folks right now. I might get in trouble for having said that. But.
Okay, well, we have just a couple of minutes. I'll give you one last question. Do you have any thoughts on why there might have been so fewer butterflies at the Volusia Sandhill compared to the Heart Island site? Or why the difference?
That's a softball for you, Cindy. You take that.
Yeah, I think the big thing is probably larval host plants. We just don't have the diversity of larval host plants at the Volusia Sandhill that's present in the Heart Island. So things like we'd see pipevine swallowtails at Heart Island. We have one measly Aristolochia plant, in the Sandhill. We just don't have that sort of neighboring area, that vast area that could supply larval host plants. I think that's huge. The things we did have, the species of butterflies we had in the Sandhill, Peter, you can pop in here if you want, were largely things like skippers, which have grass as a larval food plant.
Also a lot of Serranus blues.
Serranus blues, yeah.
Great. Well, we're at 3 o'clock, so I'm going to stop here. The questions we have remaining, we can collect them and send them to you guys if you'd be interested in giving some additional answers. Some of these we can address as well because they're asking about what to plant and how to plant. And just, I made a note in the chat, but the links that you guys provided at the end of your presentation, I threw a few of them in the chat, but we'll take all of those and put them on the webpage about this webinar, along with the recording of this presentation. So all of the attendees can look for those in about within the next 24 hours.
And this is Lisa. I just again want to say thank you to Peter, Cindy, and Karen for joining us. And don't miss our next webinar, Incredible Edible Natives, presented by Stacy Matrazo. And Stacy, that is on November 12th, is it?
10th, November 10th.
Thank you. So we'll see you then, and thank you for joining us.
Thank you.